Hi everybody, welcome to another PAG PacBio workshop. I know it's always a little chaotic to have this conference so close to the holidays, but honestly, it's the best way to start the year with the best community and one that's been so instrumental in PacBio's journey to where we are. And so with that, I'm happy to be able to talk a bit about our product roadmap before we get to the exciting research topics. So first and foremost, I do want to talk a little bit about what's coming in 2026, both on our Vega and Revio systems, and then also about the different applications you're going to hear about through the customer talks, give you a little bit of a background on what those applications are and also what is new for 2026. So first and foremost, we are a long-read sequencing company, and we have two instruments at PacBio. One is the Revio system, which is our high-throughput instrument. It's definitely open to population scale, and it runs 4 smart cells at a time. Our other instrument is the Vega. This is our benchtop machine. It runs 1 smart cell at a time and definitely fits that lower mid-throughput need. We're excited to announce, if you caught it at ASHG, that we are going to be making improvements to our SPRQ chemistry this year. This is expected mid-2026. And the exciting parts of SPRQ-Nx is what we're calling it, is that we're enabling multi-smart cell use. So what that means is that we're able to reuse a single smart cell for multiple iterations. And also it's going to help us reduce the cost of a genome down to $150 to $180 for a 1 gigabase genome diploid at 30x. So hopefully that helps you guys give a frame of reference to scale that to your species of interest. If we look a little bit closer at what smart cell reuse is going to look like, currently if you have a Revio system or if you've ever loaded one, if you're loading 8 samples, you tend to put in 2 trays of smart cells as well as the rest of the reagents, and then you hit go. That's still going to be a very similar experience, except for now you're only going to have to load 1 tray of smart cells. That tray is going to do its first collection on the first 4. The instrument is going to move right into a manual wash, and then the next 4 samples are going to get loaded and run on the machine as well. So hopefully from your perspective, it's still going to be walk away, but this is just allowing us to be more environmentally friendly, allowing these reagents to go a bit farther, and also helping you guys reduce the cost to do your sequencing. But you don't have to take my word for it. We like to show you guys examples of what these new chemistries are going to look like and how they are hopefully going to perform across species. So Jeremy Schmutz is also going to be talking on this a bit later in the workshop today. He's been a part of the SPRQ-Nx beta program, and we'll be showing you some additional species beyond these. But if you remember last year, we partnered with the California Conservation Genomics Project and the UC Davis Genomics Center to get access to some of these biodiversity samples, including the happy otter that you see here and a couple other plants and animals that were represented. What we did with these samples is we pooled 1 animal, 1 plant together into a library preparation, and then we ran that sample across 2 smart cells in sync. So the first smart cell was 1 use, then it got washed and then reused again on that same smart cell. And this is a quick look at that data. So again, for a frame of reference, we expect 100 to 120 gigabases per smart cell. And you can see that both use 1 and use 2 of these 2 smart cells yielded very well. If we split these up into the different species by their barcodes, you can see that we got really decent depth of coverage for all 4 species that were represented. And if we downsample this coverage to an even amount, and we check the assembly statistics to prove that we are still getting the same data that you've come to expect from the SPRQ chemistry, you can see that in all of these instances, we're still able to phase these assemblies. They end up in just a couple hundred contigs, and a lot of times the contig N50s are getting close to even 100 megabase pairs. The one caveat here is the rice that you see here was an inbred line, so you're only going to see the 1 assembly. If we zoom in on the happy otter that we talked about last year, we have now a lot of our different chemistries across our instrument fleets and the different assembly metrics that match. And so you can see highlighted in pink here the 2 different runs that were mapped back to that SPRQ-Nx chemistry. So 1 iteration of the smart cell and then the second use of it. Again, in both instances, we have very high contig N50s, and the assemblies we're able to assemble and phase completely. If you're interested in looking at this data yourselves, all of these results are now available to download on our website, so you can go to pacb.com/downloads or data sets, excuse me, and you will be able to see them there. We also have some exciting updates for the Vega system. So currently it is not utilizing the SPRQ chemistry that the Revio system is. So we're happy to announce that that chemistry is going to be ported over to the Vega system. This is particularly exciting because that allows us to drop the input amounts needed for sequencing from 2 micrograms down to 500 nanograms for this sequencer as well. And the update that's coming to both systems is we are continuing to expand our multiomics portfolio. So now the instruments are also going to be able to call 5-hydroxymethylcytosine as well as 5-MC and 6-MA right on the instrument itself. And so hopefully maybe you insect people in the room will be excited about that. Now I want to shift gears and move the talk a little bit into applications. So the first application I'm going to talk about today is CiFi. For those of you that came to the workshop last year, if you remember, this is a protocol that was developed by the Megan Dennis Lab out of UC Davis. And Scott Guide from the USDA talked about using this for insects, which Shanna will talk about here in a bit as well. But what CiFi is it's high-C done on our technology instead of on short read. And so this is beneficial for a couple reasons. First and foremost, it really helps us improve phasing and scaffolding because the multi-KB reads of HiFi themselves can actually have several interactions or concatenated segments on a single HiFi read, as opposed to the mate pair situation that you're used to with the short read technology. It's also able to use much lower input, so this pairs nicely with our Amplify protocol, so you can get away with fewer cells or fewer nuclei going into the prep when you utilize CiFi, again, depending on how you want to do the upfront transformation. So this is what a CiFi read looks like. You know, again, you can see the CiFi read in green down here at the bottom, and you can see all the different interactions or where that's lining up on a single chromosome. And so if we zoom in a little bit further, another benefit that we're seeing from this is even if we look at the interchromosomal pairwise distances, you can see in brown, this is what you would historically expect from the short read data. And in the green and the orange, this is the CiFi data. And if you look all the way out to the right, that's about 120 megabase pairs. We're still seeing different pairwise that are from the same chromosome. And for this year, we wanted to take this one step further. So we're introducing a workflow that we're calling single library chromosome-scale assembly, or SLiC. And what this is we're actually keeping the HiFi and the CiFi library separate until right before library preparation. And then we pool those two types together before library prep, and they can be sequenced on a single Revio cell. So for this experiment, we partnered with Megan Dennis Lab again, and we did some vol samples, and we pooled the HiFi libraries at about a 90% molarity and the CiFi library at 10% molarity and sequenced these on a single Revio cell. So again, these are the two species that were done as part of this experiment. You can see the metavol and the perivol there. And again, what we're happy to report is that using HiFi ASM as well as the YAS scaffolding that you may be familiar with this type of work, you can see that we were able to get both haplotypes here, and that also they were in the number of scaffolds that we would expect for this species. If we take a look at the perivol as well, you can see that we have very similar results, but you also see a couple false joints here. That's because these are uncurated assemblies, and we're also happy to say that the Dennis Lab just came out with the curated assemblies for these, and those false joints were very easily fixed in the curation. Another exciting component of these assemblies is that there were 2 additional circularized contigs that were about 16 kb in length, and these actually represent the mitochondrial genome, and both of these only differed by 2 SNPs, but the HiFi technology and the CiFi technology was able to still see these. So again, if we try to add some pricing to this workflow, where we're pooling these together on a single run, you only need to do 1 library preparation and 1 sequencing interaction to be able to get this SLiC assembly. And the total PacBio reagent cost for that 1 gigabase genome today is about $490 on SPRQ, and we're expecting that to be reduced down to about $350 on the SPRQ-Nx chemistry. One caveat here is that this does not include the high-C or the 3C portion up front, so make sure you're factoring that into your calculations. Okay, switching gears a little bit. We're going to talk a little bit about Fiber-seq, which I know you've heard about in past presentations as well. And Christine is going to do a deep dive that here later on in the workshop in Maze. And so what Fiber-seq is it's a way for us to get at the underlying chromatin architecture of your DNA or of your species. And so the way that we're able to achieve this with PacBio is that we can call 6MA on our sequencing technology, and you use a methyltransferase to go in and add that epigenetic modification to all of the A's that are existing in an open chromatin structure. So what happens is when you go through sequencing, we're able to see which of those actually got the methylation added, and that gives us an assessment of the underlying chromatin architecture. We've also partnered with EpiCypher to make this much more available and a kid solution. So they're here at PacBio, or excuse me, at the exhibitor hall. If you want to learn more, stop by their booth. But they have developed essentially a treatment for Fiber-seq where you take the nuclei that you've extracted, you do the Fiber-seq reaction from EpiCypher, and then from there you can extract DNA and go right into our library prep process. So again, you can get this Fiber-seq information without having to do additional library preparations or additional sequencing runs. And the exciting part of this is now if you take a look at the right, you can see all of the multiomic data that you can now get from a single sequencing run. So not only are you going to be able to phase this assembly, but you can get all variant types, whether it's SNPs, indels, or larger structural variations. You can see the 5MC methylation profiles that are haplotype specific, and now you can also see the chromatin accessibility or the chromatin architecture as well. And all these data points layered on top of each other really start showing you some amazing things, and I'm excited for you guys to see that in Christine's talk. Okay, next, Jonas did a good job of introducing this, but we did want to circle back to Kinnex. So again, we're excited that Kinnex is really helping us scale the amount of RNA reads that we're able to get out of our technology. And we're also going to introduce some tools that have been updated to help you guys do annotations more easily. So for those of you that aren't familiar with Kinnex, it's our way to concatenate multiple iso-seq reads together. Iso-seq reads are what we call our RNA-seq reads. They just happen to be full length. So we see the poly A tail, the 5' and the 3' ends. And when we pair that with Kinnex, we're concatenating these molecules together to build out a HiFi read that is 15 to 20 kb in length. Because as we know, most isoforms tend to sit in that 1 to 2 kb range. So there's a lot of real estate still to build out a HiFi read. We also partnered with the USDA, so this is Tim Smith and Kirsten Kuhn from the Clay Center location. Now that we can have such deep a number of reads per tissue type for annotations, we've also moved our benchmark up to 10 million reads per tissue is where we start seeing saturation. But again, we wanted to test this across the tree of life. So the USDA helped us do this in giraffe and red muntchuk that you see here. And in both of these instances, they found that the isoform and the genes, in terms of discovery, do both start leveling off at around that 10 million read benchmark. So we feel that this is a pretty safe read depth for you to go to as a benchmark. And again, Brooke from Corteva will go into this in more detail as well. EGAP-X has also been updated to take iso-seq reads, so this is definitely a big development from last year at PAG at this time. And as of a couple weeks ago, it can actually take iso-seq reads in two forms. So if you have the short-read RNA-seq data, you can input both that and iso-seq, or if you only have the long-read data, you can utilize that as well for the annotations. And Shanna will go into this in more detail as well. And lastly, I wanted to touch a little bit on long-read low-pass, because this has definitely been an emerging application for us. So for those of you that were at last PAG, Kendall Lee came out with this landmark publication right around that same time frame. And some of the takeaways from this publication in their work in Peanuts at the time is that if you do HiFi read as well as short read, and you do that at about a 1 to 2x depth per haplotype, what you find with the HiFi reads is that it does a much better job of representing the genome space as well as the gene space. And with the HiFi data, you're also able to maintain all structural variant types, so SNPs, indels, and larger structural variations. And this paper goes on to connect the dots that it's those large structural variations that exist in the gene space that have a much higher likelihood of affecting a trait of interest, as opposed to just the SNPs that are commonly used in a lot of these breeding programs. So again, this kind of really laid the groundwork for looking at long reads, especially at the discovery aspect for a lot of these programs. And if we look at that in a little more depth, because you might be thinking, hey, 1x seems a little bit low, what if I want to go higher? This paper also does talk about that at about 4x depth of coverage with the HiFi reads, you are starting to have at least 1x of coverage across the whole genome. So, and that's about what they need to really do this type of information. If you compare that to the short reads that you see here, at about 4x depth or at about equivalent depth, the most that we're seeing from some of these species was capping out at about 50x coverage, or excuse me, 50% coverage of both the gene space and genome. And I know you're thinking, like, Jake, that's great, but how are we going to scale PacBio's technology to meet the demands of those very high throughput applications like genotyping or like long read low pass? And so we are also happy to announce, and this is not just a PacBio effort, but across our PacBio compatible program, that from tissue disruption through sequencing, we have now created an ecosystem where you can take a 96-well plate for most species, and you're able to do this in a 96 plate format. And if we take a look at a lot of the different protocols as well as a lot of the different PacBio compatible partners that it takes to make this happen, you can see a lot of those here that we've partnered with, not only to develop automation protocols, but also to do the upfront extraction and shearing in a plate format. And talking a little bit more about that, another kit that came out this year that we've partnered with Sequel to help them test and develop is the Sequel Longplex kits. And these can also be utilized to do a lot of these high throughput applications like microbial WGS or long read low pass. And what we wanted to demonstrate here is that you're still getting equivalent coverage of the genome to the HiFi kits themselves. So this experiment, we took a control rice DNA that you see here, and we put that through the standard HiFi prep, and then we utilized the Longplex PCR kit to do 24 replicates of this same species. We index those together into a library and sequence those on a single SMRT cell. And if I back up a little bit, the exciting part about the Sequel kits is that they use a transposase-based method to do the shearing, and then they can actually do a barcode ligation at that point. So this now opens up you being able to pool samples before library preparation, and that really helps you dilute the cost of that portion of the workflow across your samples now, as opposed to having to do library prep for each individual sample. And so when we go to demux this data and look at it, we do get really good assemblies for all of the different controls that we're seeing here. And the graph on the right shows the demux was very uniform across the 24 samples that we tested. This kit can go up to 96, so 24 is not the minimum that you can do. You can scale this all the way up to 96, and if you get creative with how you do the PacBio library preparation, you can scale even further beyond that. So now we're happy to say that for these very high throughput applications, there are 3 workflows that you can really choose from depending on your application need. So first one is standard HiFi. You would use this in those instances where you want to maintain your methylation information and a lot of other information that can come from that data type. And then the Sequel kit as well as the Amplify, these both have PCR involved, and because of that, we're able to add barcodes to the samples before library preparation, so that helps reduce the costs to really bring that down if cost is important for your application. And with that, I want to thank you for your attention today. I'm going to hand it back to my colleague Jonas, and we'll get to the speaker talks. Thank you so much.